Dear students, the topic to be discussed today is microarray techniques. So, first we have to go with what is array. So, an array means it's an orderly arrangement of some samples or some components where matching of known and unknown samples is done based on a certain parameter. Here in case of microarray, mostly we will focus on DNA microarray technique in this discussion. So microarray, in case of DNA microarray technique, is a pattern of single-stranded DNA probes, means small fragments of single-stranded DNA, which are immobilized on a surface. And that particular surface is known as a chip or a slide. It may be wool surface, metal surface, or it may be glass surface. So better to use a metal surface because absorption or immobilization will be better there. So the array of an orderly arrangement of DNA samples where matching of known and unknown DNAs can be done by the principle or by the rule, that is base pairing rule. So this technology evolved from the experiment is known as southern blotting. So southern blotting is a technique, a molecular biology technique used to detect a specific DNA sequence, say in a tissue sample or blood sample. Now we have to add a restriction enzyme to cut a sample of DNA into multiple fragments that are separated using gel electrophoresis, DNA or agarose gel electrophoresis. Then the DNA fragments that are in your agarose gel to be transferred from the gel to a membrane. So in our last lecture, as we have studied about the protein gel and Western blot or immunoblot, there we have transferred the protein fragments or the proteins from STS phage to a membrane, it may be nitrocellulose membrane or PBDF membrane. So here in southern blot, we are just transferring the DNA fragments, DNA probes from the agarose gel to a particular membrane. And this particular concept of microarray was first proposed in the 1980s by Augenrich and his colleagues. So the principle lies behind this particular technique, DNA microarray, is on hybridization between the nucleotides. So hybridization, mostly DNA hybridization, what we are going to discuss today. So DNA hybridization is generally refers to a technique, molecular biology technique, that measures the degree of genetic similarity between pools of DNA sequences. Means so many DNA sequences are there. So what are the sequences that have the similarity? We can separate those sequences from the others. So it is usually used, this DNA hybridization is usually used to determine the genetic distance between two organisms. So this particular technology is being used here for this particular technique, DNA microarray. So using this technology, the presence of one genomic or complementary DNA sequence in 1 lakh or more sequences can be screened in a single hybridization technique, single hybridization process. So this is the novelty of this particular technique. Means in a one go, in a particular single experiment, so you can check whether the similarity is there or not between the DNA fragments of the DNA sequences, where the sample size may be 1 lakh or more DNA sequence. So you can imagine how many numbers of DNA sequence is present there, and we can nullify, and we can better to say we can specifically say, okay, these are the DNA sequences that are similar, have similarity in between. So how to perform this particular process? So this process possesses 7 different steps. The first step is sample preparation. If you are trying to isolate, trying to perform DNA chip assay or DNA microarray, so you can do that by using simply large cells, or else you can go for cell culture. So any kind of normal or cancerous cell or any viral infected cell, to check that, first you have to culture the cell. If it is blood, then you have to screen the blood cells you are looking for, and then you have to purify it, then you can use it. So I have considered here the cultured cells, adherent cells, so the cells to be cultured, and the full grown cultured cells to be washed using PVS, 1x PVS, multiple times to remove the traces of medium present there. And then you have to go for lysis of the cell pallet to get the messenger RNA from the cells. So we need messenger RNA from the cells, and then we have to go for cDNA synthesis, means reverse transcription from mRNA to cDNA. So the first step is sample preparation, second step is purification, and the third step is purification of what? Purification of mRNA. The third step is uh, reverse transcription. So we are getting now the complementary DNA from the mRNA that we have isolated in step 2 and isolated and purified in step 2. Now the cDNA should be labeled using some fluorophore. We have to tag this cDNA with some fluorophore, that may be CY5, CY3, like that. It has some fluorescence, and then we'll go for DNA hybridization. So this DNA hybridization will be performed in a particular machine, and we have to scan all the samples present in our mixture. So the data we will get should be normalized, and then it will be analyzed by using a particular software. So in a particular experiment, it's better to say in a single experiment, we can screen multiple number of DNA fragments using this procedure. So here there is a data. So how it is going on? So I have taken one example. So 2 petiplates are there. One petiplate contains normal cell, the right side petiplate, and the left side petiplate of this figure contains cancer cell. Here you can see. So what we are doing, this is a cancer cell, this is a normal cell. So what we are doing, we are isolating RNA by lysing the cells using a trizol method or whatever be the method. We have to lyse the cells, and both the experiments should be proceeded using 2 different vessels or 2 different processes. But what we are trying to get, we are trying to get cDNA from that. So 2 samples of cDNA, then we have to label. So the first sample should be labeled with the red fluorescent probe, and the second cDNA should be labeled with a green fluorescent probe. So the second one, I don't know which one is which. Which one is the cancerous one, which one is the normal one. That we know. But to confirm it's a cancer cell, so we are tagging or labeled the DNA fragments using two different fluorophore. The first one with say RITC, it's a red fluorescent probe, and the second one, the normal one with a green fluorescent probe, say FITC. Then we have to go for hybridization. Means we have to combine the genetic fragments or DNA fragments. Then we have to go for hybridization, and this hybridization will generate so much of data. And this data should be normalized and then analyzed using a software. So what will be the analysis? Here you can see. Now we have to go for hybridization, first leveling and then hybridization. Mix it and then drop it in different probes or in a metal surface containing different RNA fragments. And now we have to scan it. So in our laser scan, what we will get? We will get different signals. We will get different color. Each and every color will signify a different phenomenon of the DNA after hybridization. So here you can see that in case of the gray spot means not present in cell, the DNA not present in cell. In case of yellow spot, present in both cells. Means the DNA fragment present in normal cell as well as cancer cell. And when it is gray, means both the type of cells doesn't have this particular DNA fragment. But the significant data is that the red and the green one signifies that in normal cells it contains, and the red one signifies in disease cells or malignant cell or tumor cell it contains. So from this data you can see that you can screen several type of DNA, and also you will get the similarity between the DNA fragments. So what are the types of DNA microarray or simple microarray? So it may be DNA microarray. So I'm going to discuss it elaborately. cDNA microarray and oligonucleotide arrays. Next one is protein microarray. It can be of three types: analytical, functional, and reverse phase. Then chemical compound arrays. Chemical compound, a collection of organic chemical compounds spotted on the solid surface using this technique. Next one is here you can see the protein array. So in case of protein array, we have to immobilize an antibody, a native protein, and a peptide. And then we have to block the non-reactive site of the protein. Then you have to incubate the sample with some antibody tracer molecules. And after incubation, you can see specific type of binding. And this specific binding can be determined using either some tools that may be mass spectrophotometry, that may be surface plasmon resonance spectra, or you can go with atom force microscopy. Here, these are for the non-level one. Those who are non-level, so that should be determined using these techniques. But who are the leveled one? And if you have leveled proteins with the fluorophore, then you can go with confocal microscopy, or you can go with fluorescence microscope. So this is how you can check the similarity or dissimilarity between the proteins. So it will give you the data. Next is carbohydrate arrays. So various oligosaccharides or polysaccharides immobilized on a solid support, mostly the gold chip, in a spatially defined arrangement can be assessed using this technique. And the last one is cellular microarrays. So it is potted with varying materials such as antibodies, proteins, lipids, which can interact with cells, leading to their capture on specific spots. It is present there in chip. So mostly in case of carbohydrate microarray techniques, the glycan binding proteins are attached to the surface of the plate or of the chip, and glycan binding proteins are that have the clinical significance will bound with the glucose subunit. So then we can tag it with using some fluorophore where FITC has been used, and obviously multiple probes you can use. So it will give you the spots, and by interpreting the spots or analyzing the data, you can confirm that okay, this is actually the glycan moiety that has specificity for this particular type of protein. So in case of lectin analysis, in case of antibody analysis, antigen analysis, uh, in case of virus antigen, mostly they are glycoproteins. So the glycan moiety is there. So analysis of this type of pathogens or lectin proteins can be performed using, and also homology of those proteins can be analyzed using carbohydrate microarray technique. Now elaborately we will discuss about the DNA chip or the DNA microarray technique. So as I said that these are the two types. One is a CNA based and one is a oligonucleotide based. So first one is a CNA based microarray technique. And this particular type of technique or chip based assay prepared by using complementary DNA. So this particular type of assay can be called as DNA chip assay or cDNA microarray or probe DNA microarray technique. So here complementary DNAs are amplified by using polymerase chain reaction. Then it is immobilized on a solid support made up of nylon filter or of glass slide. A small volume of this DNA preparation is spotted on solid surface, making physical contact between these two. So DNA is delivered mechanically or in a robotic manner if numerous samples are there. So manually it's not possible. So you have to go with a robotic manner. When one DNA spotting is done, the pin is washed and loaded with fresh DNA to start the second cycle and to reduce the contamination. So this is how cDNA based microarray can be done. So here you can see first you have to prepare cDNA using reverse transcription process. And if you are trying to isolate it from some cells, then you have to lyse the cell, get the mRNA, go for cDNA synthesis, and then the cDNA will be mixed with another type of cDNA. You have to go for hybridization using this DNA chip. Now two type of lasers are used. One will show you the green light, one will show you the red light, and you have to combine it. By combining using some software, you can get this type of picture. So the red shows that the proteins are, the DNAs are upregulated. The green shows that DNAs are downregulated, and the black shows that there is no such expressions there. This constitutive expression is there. So which one is upregulated in case of, say, cancer, a particular type of cancer? What are the genes that are upregulated? What are the genes that are remain downregulated? In a cancer patient, we can check it, and also we can predict whether a normal person can have the probability, means the probability of having this particular type of disease. So that should be analyzed using this particular technique. Second one is the oligonucleotide based microarray technique. So here oligonucleotide techniques are probes are short sequences designed to match part of the sequence of interest. And these are produced by printing short oligonucleotide sequences designed to represent a single gene by synthesizing the sequence directly onto the array surface instead of depositing uh, in intact sequences. Now, what are the applications of microarray technique? Microarray technique has a vast application, well-versed application. So, in case of gene expression profiling, you can use it. In case of comparative genomics pool, you also can use it. In case of disease diagnosis, in case of cancer, you can use it. In case of drug discovery, and in toxicological research, microarray technique, different types of microarray techniques can be used. Now, what are the advantages of this technique? The most advantage of this technique is that it provides data for thousands of genes. Well, although we have to perform only one experiment instead of many. And we are getting the data very fast and easy. So huge step closer to discovering cures for diseases and cancer. So in a single experiment, you can analyze homology or similarity between the genes or the fragments, DNA fragments that are the culprit fragment DNA genes or the genes that are actually responsible for causing cancer, and also the homology or similarity between the genes present in the normal cell. So huge step closer to discovering cure for diseases of cancer and cancer. And different parts of DNA can be used to study gene expression. No specificity is there. But there are some disadvantages. The biggest disadvantage is DNA chip assay is a very expensive assay, very costly. Production of too many results at a time requires a long time analysis, which is quite complex in nature. And only experts can analyze this, not the layman. So this is indeed a quite indeed a complex process. This particular chip assay do not have very long shelf life, as chips do not have very long shelf life, so we cannot keep it for long, for months, for years. So that proves to be another major disadvantage of this particular technology. These are the references from where I have developed. I have taken some information to develop this lecture. So you can also go through these books for better understanding and also for other references. Thank you.